One of the biggest mysteries in human history has always been this: where did we come from? How many human species walked the earth before us? And what is our real connection to them? For centuries, the only way to answer this question was by studying the shape of bones, their size, their joints, their proportions. This method can tell you a lot, but it also hides a lot. Bone shape can tell you whether a creature was tall or short, whether its brain was large or small, but it cannot tell you who it mated with, who it mixed with, or whose blood ran through its descendants. But there's a basic fact that anyone interested in this field needs to understand. DNA does not last forever. It is a chemical substance that keeps breaking down over time, especially in the presence of heat, moisture, and bacteria. It is not like a rock or a bone that can sit unchanged for millions of years. So whenever a headline claims that scientists extracted DNA from a fossil that is such and such many 100,000 years old, the first question should always be, is this even possible? Answering that question matters, because the real story, the one I'm about to tell you, is already remarkable enough on its own. It doesn't need any exaggeration, and unlike many viral claims that circulate online, every discovery in this article comes from peer-reviewed research, published in major scientific journals conducted by named researchers at named institutions, whose work has been checked, challenged, and confirmed by other scientists around the world. That distinction between a headline built to shock and a finding that survived scrutiny is the whole point of this story. Scientists have spent decades studying how fast DNA breaks down, and they've learned that the answer depends heavily on the environment it was preserved in. Warm, damp, or exposed conditions can make DNA unreadable within just a few thousand years. But extreme cold, like permafrost, the permanently frozen ground found in Siberia, Greenland, and other polar regions, can extend that window dramatically. Why does cold matter so much? DNA is a long molecule, and over time, water and enzymes slowly cut it into smaller and smaller fragments, until eventually there's nothing left to read. Heat speeds up these chemical reactions. Moisture accelerates them further because water is often the medium through which the damaging reactions occur. Bacteria and fungi make things even worse, since living microorganisms actively break down organic material as part of their own survival. And a warm, wet environment is exactly where microorganisms thrive. Put a bone in a hot, humid climate, and within a few thousand years, sometimes less, the DNA inside it is reduced to fragments too short and too damaged to tell scientists anything useful. Cold does the opposite. It slows down every one of these chemical processes. In permafrost, temperatures stay below freezing year-round, sometimes for hundreds of thousands of years without interruption. This means the DNA trapped inside frozen bones, teeth, or sediment barely reacts at all. It's a bit like keeping food in a freezer instead of leaving it out on a counter. The same food lasts vastly longer under refrigeration, and ancient DNA works on the same basic principle, just stretched across geological time rather than a kitchen's worth of days. This is why almost all of the oldest preserved DNA ever recovered, whether from animals or humans, comes from these frozen environments. That's not a coincidence. It's basic chemistry. In 2022, a team of scientists extracted DNA from sediment layers buried in Greenland's permafrost, dating back roughly 2 million years, the oldest DNA ever recovered at that time. This came from a site called Copenhagen Formation in northern Greenland, and the discovery was published in the journal Nature. But here's something important to understand. This DNA didn't come from a fossil, a bone, or any human remains at all. It was environmental DNA. Tiny fragments scattered through soil and sediment, left behind over centuries by animals, plants, and microorganisms, as they lived, died, and decomposed in that landscape. This DNA revealed that the region was once a lush forest, home to mastodons, and an entire ecosystem of ancient plants, insects, and microorganisms that no longer exist in that form today. Researchers identified traces from more than 100 different plant genera, along with animals, including reindeer, hares, and even mastodons, a type of ancient elephant relative that scientists previously had no direct evidence had ever roamed that far north. It painted a picture of the Arctic as it existed 2 million years ago, a place with a much warmer climate than today, filled with a mixture of Arctic and temperate species living side by side. But this DNA couldn't tell us anything about a specific individual or specimen. This distinction matters a great deal. Environmental DNA gives us a picture of a place's past, what kinds of life existed there, roughly how abundant certain species were, what the ecosystem looked like as a whole. Fossil DNA gives us a direct, specific answer about a particular creature, a particular body, a particular lineage, and how that individual relates genetically to other individuals, other populations, and other species. And in the world of fossil DNA, the actual record doesn't reach nearly as far back as the environmental DNA record does. That gap between the two records is exactly where a lot of public confusion comes from. And it's exactly where sensational, inaccurate headlines tend to take root. If you ask where the most significant DNA discovery in human history took place, the answer is inside a cave near the town of Atapuerca, Spain, known as Cima de los Huesos, meaning the pit of bones. This cave has been a treasure trove for paleontologists for decades. Hundreds of human-like bones have been recovered here, the remains of at least 28 individuals, which scientists have attributed to Homo, heidelbergensis, or a closely related older species. Excavations at the site began in earnest in the 1980s, led by Spanish paleoanthropologist Juan Luis Arzuaga and his colleagues, and over the following decades, the pit yielded one of the richest collections of archaic human fossils anywhere in the world. Skulls, pelvises, limb bones, and even a complete set of teeth from multiple individuals, all apparently accumulated at the bottom of a natural shaft deep inside the cave system. These bones have been dated to roughly 430,000 years old, based on a combination of uranium series dating and other geological dating techniques applied to the surrounding sediment layers. In 2013, a team from the Max Planck Institute for Evolutionary Anthropology, led by the renowned geneticist Svante Pääbo, attempted to extract DNA from these bones, and succeeded. This was, at the time, the oldest human DNA ever recovered, and it was no small achievement. A span of 430,000 years is so long that before this, no scientist believed any usable DNA could be extracted from bones that old. Pääbo himself later described the result as something his team had barely dared to hope for, given how degraded ancient DNA typically becomes over such a long stretch of time. What makes this discovery even more remarkable is that this cave was not some frozen permafrost-like location. It's a limestone cave in Spain, a country with a temperate, not Arctic, climate. But its depth, constant coolness, and unique conditions preserved the bones so well that far more DNA survived inside them than anyone had previously imagined possible. The cave sits far below the surface, shielded from daily and seasonal temperature swings, and the specific chemistry of the sediment surrounding the bones appears to have helped protect the genetic material trapped within them. Extracting this DNA wasn't simple. The team had to work in special clean room laboratories, wearing full protective suits, because ancient DNA exists in such tiny, degraded quantities that even a stray skin cell or a breath from a researcher could contaminate a sample with modern human DNA and ruin the results. Scientists in this field describe ancient DNA work as being closer to forensic science than typical laboratory biology. Every possible source of contamination has to be controlled for, and results are typically cross-checked using multiple independent samples and sometimes multiple independent laboratories before they're considered reliable enough to publish. The initial results revealed something surprising and at first confusing. The small amount of mitochondrial DNA they managed to extract was more closely related to the Denisovan lineage, even though the shape of the bones resembled Neanderthals. Mitochondrial DNA is a small, separate piece of genetic material found outside the main nucleus of a cell, and it's often the easiest kind of ancient DNA to recover, because each cell contains many more copies of it compared to nuclear DNA. This puzzle raised a new question for scientists. Was the relationship between Neanderthals and Denisovans more tangled than we'd assumed? Had there been an earlier, more ancient population that contributed to both lineages in ways not yet understood? Later, once the team used improved techniques to extract a full nuclear genome, a much harder task, since nuclear DNA is far more degraded and exists in far smaller quantities in ancient bone than mitochondrial DNA, the picture became clear. These individuals were early ancestors of the Neanderthal lineage. This discovery became key evidence that the split between Neanderthals and modern humans occurred somewhere between 400,000 and 500,000 years ago, an estimate that had previously been based only on bone shape and rough calculations of mutation rates, but was now backed by direct genetic evidence extracted from a population that lived right around the time of that split itself. The other side of the Cima de los Huesos story, the Denisovans, is its own remarkable tale, and it began several years earlier in a completely different part of the world. In 2010, a small fragment of bone was found in Siberia's Denisova cave, just a finger bone, so small that examining it alone offered no real clues about which species it belonged to. The cave itself had been known to archaeologists for a long time and had already produced stone tools and other artifacts, suggesting it had been used by ancient humans over an extended period. But nothing about the fragment's size or shape gave any hint of what was about to be discovered. When DNA was extracted from it, the results stunned scientists. It wasn't modern human, and it wasn't Neanderthal either. It was an entirely new, previously unknown human species, now called Denisovan, named simply after the cave in which the bone was found. This was perhaps the first time in history that a human species was identified purely through DNA, not through a complete skull or skeleton, not through careful measurement of bone proportions, not through comparison with known anatomical features, just one. Small bone and the power of DNA told us a story that had been hidden for hundreds of thousands of years. In the years since, additional Denisovan fossils have been found, including a jawbone discovered in Baixia Karst Cave on the Tibetan Plateau in China, identified in 2019 through analysis of ancient proteins preserved within it, a technique that complements DNA analysis and can sometimes work even when DNA itself has degraded beyond recovery. Later research also revealed that the Denisovan legacy isn't confined to history. It's still alive in us today. In modern humans, particularly people from Melanesia and Papua New Guinea, Denisovan DNA makes up 4 to 6% of their genetic makeup. In Tibetan populations, a specific Denisovan gene, known as EPAS1, has been found that helps people survive in low oxygen, high altitude environments, meaning that a genetic gift from a long extinct species is still saving lives today, helping people living at extreme elevations regulate their blood oxygen levels more efficiently than populations without this gene variant. This finding is one of the clearest examples scientists have of what researchers call adaptive introgression, a case where genetic material from an extinct human relative didn't just get passed down by chance, but actually provided a real survival advantage that natural selection then favored and spread through a population over thousands of years. It's a striking illustration of how interbreeding between different human species didn't just leave behind harmless genetic traces. In some cases, it left behind tools that helped modern humans survive in environments they might otherwise have struggled to inhabit. Now, let's return to the basic question we started with. If the 430,000-year-old DNA from Cima de los Huesos is the oldest fossil DNA ever recovered, how could a claim about DNA extracted from a 900,000-year-old fossil possibly be true? The answer is straightforward. This hasn't happened so far, and based on current science, it would be extremely difficult right now, especially in an environment that isn't as cold as permafrost. The 430,000-year success at Cima de los Huesos was already so extraordinary that scientists called it astonishing and beyond expectations. DNA degradation accelerates so dramatically with every additional 100,000 years that a span of 900,000 years, more than double the Cima de los Huesos timeline, is currently considered essentially impossible for human DNA to survive, especially outside a permanently frozen environment. Researchers who study the mathematics of DNA decay have proposed rough estimates for how long DNA could theoretically survive under ideal constant freezing conditions. Some models suggest a theoretical outer limit somewhere in the range of 1 to a few million years under the most ideal permafrost conditions imaginable, with essentially every variable working in the DNA's favor. But those theoretical estimates are for the absolute best-case scenario, constant sub-zero. Temperatures, minimal water exposure, and no disturbance for the entire span of time. A typical fossil site, especially one in a temperate or warm climate, falls enormously short of those ideal conditions. This is exactly why real-world recoveries of usable fossil DNA lag so far behind the theoretical maximum, and why 900,000-year-old fossil DNA from anywhere other than the most extreme frozen conditions remains outside what today's science can actually achieve. That's why, whenever an article or video claims that scientists extracted DNA from a 900,000-year-old human fossil that changes everything, that claim goes against current scientific understanding. It hasn't been published in any credible research journal, and no credible scientist makes this claim. Claims like this tend to spread because they borrow the language and structure of real scientific reporting. Specific numbers, dramatic framing, references to researchers and findings, without any of the underlying substance, no named research team, no published paper, no institution that can be checked, and no data that other scientists can examine or challenge. Here's another important example that helps illustrate just how difficult extracting fossil DNA really is. In South Africa's Rising Star Cave system, bones belonging to a species called Homo naledi have been found, dated to roughly 236,000 to 335,000 years old. This species is considered highly significant in paleoanthropology because of its small brain size and mix of primitive and modern physical traits, a combination that surprised researchers when the species was first formally described in 2015 by a team led by paleoanthropologist Lee Berger. The discovery of Homo naledi was itself a remarkable story of exploration. The bones were found deep within an extremely narrow and difficult-to-reach cave chamber, accessible only through tight passages that required specially built teams of slender. Researchers, sometimes recruited specifically because of their small body size, to squeeze through gaps barely wide enough for a human body. The sheer number of bones recovered from this single chamber, representing at least 15 individuals, raised intriguing questions about how they came to be there, with some researchers proposing that the chamber may have been used deliberately for the disposal of the dead, a behavior that would be remarkable for a species with such a small brain. But despite the wealth of skeletal material recovered, Africa's warm climate is far from ideal for DNA preservation. Heat and humidity break down DNA so quickly that, despite extensive effort, no usable DNA has ever been recovered from any Homo naledi bone. Even though this species is, in some respects, more recent than the Cima de los Huesos remains, this example makes it clear that DNA survival depends just as much on environment as it does on age. A younger fossil in a hot climate can be far less useful for genetic study than an older fossil preserved in a cold one. In 2010, Svante Pääbo and his team accomplished another landmark feat. They successfully sequenced the complete Neanderthal genome, drawing primarily on bone samples recovered from Vindija Cave in Croatia, along with other Neanderthal remains from across Europe. This is considered one of the most important scientific achievements in human history, and it was this very body of work, along with his broader contributions to founding the field of paleogenomics, that earned Pääbo the Nobel Prize in Physiology or Medicine in 2022. Pääbo's career had, by that point, spanned more than 3 decades of pioneering work in extracting and analyzing ancient DNA, starting with early attempts on Egyptian mummies in the 1980s that were later found to be contaminated with modern DNA. A humbling early lesson in the field about just how easily ancient samples can be compromised. Those early setbacks helped shape the strict contamination control protocols that define the field today, and without them, discoveries like the Neanderthal genome and the Denisovan fingerbone likely would not have been possible, or would not have been trusted by the wider scientific community, even if they had been announced. This research answered a question that had lingered for centuries. Did modern humans and Neanderthals interbreed? The answer was yes. The results showed that most people whose ancestry traces outside of Africa carry roughly 1 to 2% Neanderthal DNA today. This means that when our ancestors left Africa around 60,000 years ago and spread across Europe and Asia, they encountered Neanderthals, and not just encountered them, but interbred with them, leaving a genetic signature that persists in living populations tens of thousands of years later. This discovery wasn't just an academic footnote. It reshaped how humans understand their own identity. Before this, Neanderthals were largely viewed as a failed or inferior species that simply died out in the face of modern humans, sometimes even portrayed in popular culture as slow, witted, or unsophisticated. Now we know that Neanderthals live on inside us, literally in our genes. Subsequent research has linked specific Neanderthal gene variants to a range of traits in modern humans, including aspects of immune response, skin and hair, characteristics, and even, according to some studies, factors that influence sleep patterns and susceptibility to certain diseases. Some studies have also suggested that specific Neanderthal-derived genetic variants may have influenced how severely different individuals were affected by COVID-19, a finding that underscores just how directly ancient interbreeding continues to shape human health today. The success at Cima de los Huesos and the sequencing of the Neanderthal genome were not isolated events. They were part of a broader explosion in the field of ancient DNA research that has unfolded over roughly the past 15 years, driven by rapid improvements in sequencing technology and extraction techniques. One especially important milestone came from the genome of a young boy known as the Ust-Ishim individual, whose remains were found along the banks of the Irtysh River in Siberia. His genome, sequenced and published in 2014, was dated to approximately 45,000 years old, making it one of the oldest modern human genomes ever recovered. And what made this genome so valuable was that it allowed researchers to calculate, with much greater precision than before, the approximate timing of when modern humans and Neanderthals interbred, pointing to a window roughly 50,000 to 60,000 years ago, consistent with the early period of modern human migration out of Africa. Ancient DNA research has also transformed our understanding of population movements within more recent human history. Studies drawing on ancient genomes from across Europe and Asia, championed in particular by researchers such as David Reich at Harvard Medical School, have revealed large-scale migrations that were previously invisible to archaeology alone, including a massive movement of Bronze Age herders from the Eurasian steppe into Europe, roughly 5,000 years ago, a migration that appears to have dramatically reshaped the genetic makeup of the European continent and is now linked to the spread of Indo-European languages. In the Americas, the genome of a roughly 12,600-year-old infant known as the Enzic child, found in Montana and published in 2014, provided some of the strongest genetic evidence linking ancient Native American populations to their ancestors who crossed from Siberia into North America via the Bering Land Bridge. Similarly, the genome of a young woman found in an underwater cave system in Mexico, nicknamed Naya and dated to roughly 12,000 to 13,000 years old, offered further confirmation of these ancient migration patterns, recovered under extraordinarily difficult conditions by cave diving researchers. Ancient DNA has also reshaped our understanding of more recent history. The genome of Cheddar Man, a roughly 10,000-year-old skeleton found in England, revealed in 2018 that early inhabitants of Britain likely had dark skin and blue eyes, a combination that surprised many people and offered a striking reminder that traits we associate today with specific regions were not fixed throughout history, but shifted over thousands of years as populations mixed, migrated, and adapted to new environments and diets. Ancient DNA research has even reached into the history of disease itself. Scientists have successfully extracted and sequenced DNA from the bacterium responsible for the Black Death, Yersinia pestis, from the teeth of plague victims buried centuries ago, allowing researchers to trace the pathogen's evolution and spread across medieval Europe with a level of precision that historical records alone could never provide. This kind of research has expanded beyond human ancestry into an entirely new field sometimes called molecular archaeology, where ancient pathogens, plants, and animals are all studied using the same fundamental techniques, developed originally for studying ancient humans. It's worth pausing to explain, in plain terms, what this process actually looks like in a laboratory, because the rigor involved is a large part of why these discoveries are trustworthy in the first place. Ancient DNA typically survives only in specific parts of a skeleton, most commonly dense bones like the petrous portion of the skull, located behind the ear, which protects DNA remarkably well due to its density, and in teeth, where the hard enamel and dentin similarly shield genetic material from the environment. Researchers drill or extract a small sample from these areas, taking great care to avoid handling the sample with bare hands and often working inside dedicated ancient DNA clean rooms that are physically separate from any laboratory that works with modern DNA samples to prevent any possibility of cross-contamination. Once extracted, the DNA is processed and sequenced using specialized techniques designed to handle degraded, fragmented material, since ancient DNA rarely comes in long, intact strands the way fresh DNA does. Modern sequencing technology can piece together millions of tiny overlapping fragments and reconstruct them into a coherent genome, a process that would have been unimaginable to scientists working just a few decades ago. Because the DNA is so fragmented and present in such tiny quantities, the risk of contamination from modern human DNA, whether from researchers, previous handlers of the specimen, or even airborne particles in a museum storage room, is a constant and serious concern. To guard against this, scientists use several forms of verification. They look for specific chemical damage patterns that are characteristic of genuinely ancient DNA, since DNA degrades in predictable ways over time, and genuinely old samples show these telltale signs of age at the molecular level in ways that contamination from a modern source would not. They also frequently sequence the same sample multiple times, sometimes in entirely different laboratories, using different equipment and different research teams, specifically so that any result can be independently checked before it's considered reliable. Only once these safeguards are satisfied, and once results have gone through the formal peer review process required for publication in a scientific journal, does a finding become part of the accepted scientific record. This is precisely the kind of rigorous, independently checked process that separates the real discoveries described in this article from viral claims that appear online without any of this underlying verification. A genuine discovery comes with a named lead, researcher, a named institution, a published paper that other scientists can read and critique, and a body of supporting evidence that can be independently tested. A false or exaggerated claim typically has none of these things, no matter how dramatic or convincing its headline sounds. The biggest lesson from all these discoveries is that the human story has never been a straight line. We often imagined human evolution as a simple, linear path. One species leading to another, and eventually to us. But DNA has taught us that the reality is far more tangled. Multiple species existed on Earth at the same time. They sometimes mixed with one another, and their genetic legacy is still alive in our bodies today. Neanderthal DNA affects our immune system. Denisovan DNA helps some people breathe more easily at high altitudes. Ancient migrations, invisible to any archaeologist studying pottery or tools alone, are written directly into the genomes of people living today, in patterns that stretch from the Siberian steppe to the highlands of Tibet to the islands of the Pacific. This isn't just historical trivia. It's part of our everyday physical lives, shaping everything from how our bodies respond to illness to where our ancestors' journeys took them thousands of years before any written record existed. And the most fascinating part of this whole story is that the older something is, the harder it fights to give up its secrets. But when it finally does, the answer is always more complex and more astonishing than expected. Scientists are still searching for new methods to extract DNA from even older bones, particularly from permafrost and cold caves, and new techniques are being developed constantly, from improved methods of separating ancient DNA fragments from contamination to entirely new approaches like the extraction of ancient proteins, which can sometimes survive even after DNA itself has broken down beyond recovery. Every new sample, every new technique becomes an opportunity to illuminate a new chapter of human history that no one previously thought could be recovered. This real story, without any exaggeration, is already extraordinary enough that it needs no false claims. The journey of the human species, built on hundreds of thousands of years, several different species, and countless encounters, is just as astonishing in its true form as any exaggeration could ever make it seem. And unlike claims that collapse the moment anyone looks for the underlying research, this story only grows stronger the closer you examine it, because every piece of it has already survived exactly that kind of scrutiny.